everyone. I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas at San Antonio's neuroscience research podcast. So today is September 16th. Is that right? Oh, could you hear my computer dinging? Oops, I'll have to turn all that stuff off. I'm on a new computer. Okay. Today is September 16th, 2021, and we're talking with Max Fletcher, who is Associate Professor in the Department of Anatomy and Neurobiology at the University of Tennessee Health Sciences Center. Hi, Max.
Hi.
So Fletcher Lab studies sensory representations in the olfactory and more recently in the taste systems of mice and how they are modulated during learning and decision making. He uses a range of techniques, including in vivo electrophysiology, awakened and anesthetized in vivo imaging, in vivo 2-photon and wide field calcium imaging, as well as lots of different behavioral paradigms. So today in the Zoom, we've got our peripheral taste specialist. Not that you're peripheral, but you're a specialist of peripheral taste. Lindsey Macpherson, you're very central. Hi, Lindsey.
Hi, Salma. Thanks.
And we've got Charlie Wilson. Hi, Charlie. And Todd Troyer. Hi.
Hello.
So, okay, I'd like to focus on the taste part of this today, but do feel free to bring in whatever whatever comes up about olfaction as well, because it's very relevant too. So, Max, the three kind of ancient expectations that, four actually, that we have about primary sensory cortices are that they contain some sort of fundamental representation of a stimulus quality or of all qualities, that they're distributed in some sort of topographically meaningful way, that they serve some sort of perceptual discrimination or some sort of perceptual activity or process, and that when you lesion the area, that's four, when you lesion the area, that perceptual ability goes away. So we can dig in a lot of different places there, and I hope we will, but I'd like to kind of start with the big thing underlying all of that, which is should we even think about gustatory cortex in terms of perceptual function, right? Because that's sort of like the key question, right? And I hope, do you want to start there? I mean, yeah, well, the one thing I want to kind of add is, because in terms of gustatory cortex, it's not really like any other cortex, right? That in terms of the readout is a behavior, right? You can move through the world looking at things passively without necessarily motivating or interacting with them. But when something's on your tongue, you either have to chew or spit it out, right? There's a really clear decision that you have to make. So anyway, sorry, go ahead.
Yeah, so I think that's a great question, and sort of a question that I mean, when you go to grad school, that's sort of where you're taught, right? Like, oh, this is primary cortex, and it is the area that does the final thing, right? And so, yeah, I think that I started as an olfactory for most of the time, an olfactory person, and it's maybe more true in olfactory cortex and piriform cortex. I mean, it's one step, right? I mean, primary olfactory cortex we usually consider piriform cortex, and it's like sensory neuron, bulb, piriform cortex. So it's really early on, kind of it's not even neocortex, right? But it's really early on. So probably in those cases, it is really, sort of that kind of more maybe fits that definition of that kind of cortex, even though it's kind of weird, I think. But definitely, and so that's kind of the preconceived notion I had going into doing this stuff with John Botter, who's the other guy in our department, who's the real taste person. And so, this sort of, like as things have progressed in the past few years, just jumping in sort of on what I feel sort of the back end of it, it really seems like maybe that's, not true. But you don't need gustatory cortex. Maybe there's no reason, I guess, is the best way that it has to be making these very simple decisions on what you're tasting. And it's really weird. Like I always just jump back to olfaction, but if you think about olfaction, I mean, the number of odorants that we can detect is maybe in the millions, right? I mean, I don't even know the upper limit now. It changes all the time, but it's certainly a lot, right? But the number of tastes is not very many, right? It's a handful. So if I always kind of think about that. Maybe you don't need the system to do the things that the visual system does, or the auditory system does, or even the olfactory system does, and sort of break things apart and then put them back together into their final perception, or whatever you want to call it, that you can act upon. It's already there, and there's all these crazy things where people could show that, rats still make the face when they taste bitter, even if they don't have a cortex at all. So there's some low level ability to sort of, this is good and bad, sort of built in from the beginning. So yeah, it's been a challenge as we go forward that maybe other people think that already, but for me, I sort of didn't. I think, primary cortex should be the thing, and it should do just, it should primarily be taste identity or something. And yeah, it's becoming more and more apparent, not just from us, but a lot of people that, that's not maybe really what it's doing. And I don't know that we really can say, at least from my perspective, it's really hard to know exactly, put a thing on it that says this is exactly what it does. But that's kind of what makes it fun.
So there's two questions come to mind, which are, one you've worked on yourself, which is defining kind of the anatomical borders of what gustatory cortex is. Also the topography you've worked a lot on. And then the third question is the one about the lesions. So when we lesion the gustatory cortex, what happens? So it's
so I think, oh gosh, if I can remember. So I think maybe not a lot, right?
Lindsey Shear did some of this stuff,
yes,
I think.
Yes, and so a lot of that has been more in tune with CTA and learning. So it, puts another spin on
exactly.
it.
That's
Certainly,
the point. Yeah.
certainly, like where we're considering cortex is, you can kind of, and the lesion studies are here and there because they're so hard to really make perfect. I think we're probably at a point now where we can do more fancy versions of lesions with dreads or opto or whatever, but yeah, I think maybe not a whole lot, right? The animal still seems to be able to learn, can tell you this is good or this is bad, these kinds of things. So
it seems like there has been some question, right, Lindsey? A lot of the work from Zucker's group does talk about a topography. How much of that is, what can you talk around some of that? And it seems to me that some of that is at least based on the fact that the definition of gustatory cortex may be, I don't know, is there a specific definition, because it's kind of embedded in this insular cortex, right?
No, right. And so I think that's why coming from it from sort of an outside, that's why we really wanted to look at the thalamic projections and sort of say, where is it? Because it gets really crazy. Insular cortex is this big, long piece of cortex. And kind of as you go anterior to posterior, maybe there's this other crazy idea that I really like that maybe it's just a big area that represents internal state, right? And so anything that's sort of going on inside you can be represented in insular cortex, maybe in some kind of overlapping gradient, but more posterior parts of insular cortex start to respond to foot shocks or sickness or lots of other bad things, and then that sort of goes into sort of the Zucker point that somewhere in there, as you sort of go from way posterior, not gustatory cortex, to feed into gustatory cortex, maybe you hit this, you sort of overlap with this bad, and maybe as you go a little bit more anterior, you get into the good. And so I think we were sort of playing with the idea of, well, okay, if there's sort of just this big area, and then this is where the taste information comes in, is it its own unique thing, right? Or is it just part of another set of information that insular cortex receives about the internal state that you're in, right? And so maybe it's better to think about it in that way. I don't think up until very recently, that's not been the way that people think about it, and maybe that allows you to sort of ask questions that you wouldn't normally ask, and maybe a lot of things. I think from what we talked about today, there's this kind of idea that maybe it is about consumption, or what the animal's actually doing. It's sort of a bigger picture than just this tastes sweet or not.
So do we know anything about how complex taste perception is? How complicated and fine does it get? People have people looked that? It's hard because a lot of the the behavioral things are wrapped up with olfaction, right? So but do we know how much complex discrimination there is just in the taste system?
You mean like from like a single cell kind of thing, or a population,
or anything? Or even even behaviorally. You kind of stated, well, it's pretty simple, but how much of people looked and pushed it?
I think quite a bit, right? you can do, I mean, certainly this, the sort of experiments you would expect to be done have been done, where I can have an animal, I can give the animal the different tastes and see how the cells respond, and then ask, from this population, can I decode from that the different tastes, just like you probably would in another sensory system. And I think that's all been done, right? But I think on some level, yeah, it's harder because there's just these basic, and it becomes harder, especially when I think about it from odors. I mean, I run out of room in the number of odors. I can't give an animal 1,000 odors, and that's probably not very many in terms of what we encounter on a daily basis, right? But we all just encounter the same few tastes, right? And so
just because the number of receptors is so small, or I mean, because the number of things we taste is so small.
Yes, because the number of things we taste is like more or less a few things, right? Like how many, this is a big thing. I think there's like, Lindsey probably knows a lot more than this, but like taste receptors, how many taste receptors are there? Like different genes that encode taste receptors? 30?
Yeah,
ish.
like there's 30 bitters, or 30 some bitters, and then the rest is like 5, right?
Yeah,
and there's like
right?
2 pairs of GPCRs for the first sweet, 2 pairs of GPCRs for umami. One of them is shared. and then you've got the sour autopetrin, and then an ENaC channel in mice for salt. So the receptor repertoire is minimal.
There's still a lot of combinations. I mean, I was just trying to think about the combinations of the things that you were saying. You were acting like there wasn't a lot, but all these, you meet all these people who claim that they can tell the difference between all these different kinds of beer on the basis of the particular kind of bitter that they are.
But that's all smell.
An illusion?
Well, I think you may be able to. But bitter is an interesting one, right? Because there's all these different receptors for bitter, right? Do we know, yeah? But in smell, right? There's not a lot of, probably, like the right word, I guess, is synergy or interaction, like mixtures, right? Like if you drink something that's salty and sweet, it just tastes salty and sweet. Or if you eat something that's salty and sweet, if I give you 2 different odors and you smell them and you're like, this smells like odor A and this smells like odor B, and then I mix them in sort of an equal thing, they may not smell like, they could smell like A, they could smell like B, they could smell like A plus B, or they could smell like something completely different, right? And so I think that, yeah, there's something to that the number of possible stimuli combinations is much lower, right? And then it's kind of already segregated very early on into the system.
So that's what kind of leading. Sorry, go ahead, Charlie.
So what I was just wondering what the brain has to do. Like if it's just that everything that you can taste is already encoded at the receptor level and combinations aren't important, then the brain has no job. I mean, the brain could just basically say, when something tastes bad, I stick out my tongue.
Right, but I mean, I guess there are I mean, I guess sometimes I know I'm guilty of this. Is like we get caught up in asking these questions without really thinking about what they mean to the animal and an animal's normal behavior, right? I mean, we need to taste sweet because we need to know what's sugar for us to eat, and we need salt, right? A lot of these have to do with appetite, right? And so that's certainly circuits that are central, right? Like when I'm salt deprived, I probably have a lower sensitivity to salt. That's the right way. I'm less sensitive to salt, and I'll drink more of it, right? So there are these circuits that are built in, but they're just not identity, right? Like they're not made for knowing this is salt. Whereas I don't know that that's true, say, in the olfactory system, right? It's just a very different. I think sometimes it's hard to compare systems without thinking about what their primary sort of original job was for the animal.
Yeah, so I was thinking about it. In auditory cortex, you have a little bit of a flavor of this, right? So you can do tone discrimination without, with bilateral, just, get rid of your auditory cortex, and animals can still do tone discrimination. But they can't do like complex amplitude modulated tones. And then if you go out beyond primary cortex, there's a lot of argument about what some of these secondary auditory cortex areas are doing, right? Are they to do auditory objects? Or are they doing more like integrating audition with other stuff, right? And so maybe in gustatory cortex, that's because it's simple enough, it's already leaking into primary, or I don't know how to think about that in terms of maybe the simple discrimination is already done, and so it's mostly the integration part with other behaviors or other kinds of things.
And there is a huge, I mean, there's direct projections from olfactory cortex to insular cortex. So, you know, like, because when we eat, it's flavor, right? Like Lindsey mentioned, like discriminating beers is often done by what we call taste, but it's really not taste, it's really flavor, which is olfaction. So maybe it's really hard to even talk about just one. I mean, we never probably really experience too many tastes without a olfactory component when we eat or when they, so maybe it's sort of too simplistic to even consider it, you know, a single standalone sort of sensory system. And you need on some level to maybe, and it is, and to be fair, that there is, you know, gustatory cortex will respond to odors, piriform cortex will respond to taste. So, you know, there could be a lot of that going on there that's more about, you know, like the overall, whatever you want to call it, flavor of fooding that needs to be, that's where that's happening, right? Because I think that's probably the first, gustatory cortex is probably the first area where odor information is integrated with taste information. I think that's probably true. So, you know, there's another big component, but that may be very special to, you know, just feeding in those kinds of circumstances.
Lindsey, did you want to jump in?
So yeah, I was thinking about the, you know, taste mixers. I brought this up maybe earlier, but I mean, at least at the level of the peripheral gustatory system, if you give, you know, bitter and sweet together, you get ganglion neurons that will essentially be the bitter plus the sweet population, you know, additive. But like, is that changed in the cortex? Like how, I mean, there's the top down kind of back and forth regulation of, you know, if you have something that's bitter and sweet together, you know, kind of bitter tends to win because it is so aversive. So there's kind of like a top down, you know, inhibit the eating of this because it contains something bad, even though it contains something good, right? So it kind of like this push pull of response. But in the periphery, which, you know, I focus mostly on the periphery, it's pretty additive. So there's no like, there's no real inhibition at that point, right? So that it's happening further up. But that I was thinking for the, you know, representation within the gustatory cortex, if you have a mixture, would it be seen as something novel or as just as a combination?
Yeah,
so
no,
I think that's just
yeah, it's a great question, right? And then we really weren't thinking in that way when we did them. I was just talking to John about it, like after the talk, it's like, oh, we really need to try that. That's a neat idea. But yeah, yeah.
So
I mean, I guess.
Sorry, go ahead.
I guess sort of the way maybe this is wrong, and the way I've kind of always thought about it is that you know, like at least thinking about it from olfaction, and maybe it carries over, is that you have some straightforward circuit that processes sensory information sort of just without any other influence, right? Even probably in the olfactory system, this is this smell, this is this smell, and this is this smell. Somewhere along the way, like sort of like Lindsey was saying, there's all these other systems that can impinge upon that to sort of modulate that response depending on the state of the animal, right? You know, and so, you know, maybe you need maybe that's where cortex. The problem is I come from bulb is a strange place, right? Like for all the sensory inputs coming into bulb, there's just as much centrifugal inputs from the rest of the brain coming into bulb. So bulb is this really great, crazy place that kind of can do that, right? Like hunger, like whatever state you're in can really heavily modulate the sensory responses, especially like sensitivity to odor input, right? And so maybe that's sort of the job that these other cortical areas are doing in the other systems. I mean, olfaction is probably not ever good to compare to. Maybe olfaction is just weird in general in that way. So maybe it's hard to do, but maybe that's the point. I mean, you know that like since your preferences to food taste, your preferences to sweet, salty, whatever, change depending on the state you're in, hunger, these kinds of things, right? Or you know, neophobia is another one where an animal that hasn't experienced the taste before will be hesitant to drink it, right? Because they don't really know if it's good or bad or not, right? And maybe those kinds of things are being modulated. You have some signal that's saying this is what it tastes like, but maybe how I respond to what it tastes like, it's different depending on the state that I'm in. I think that's sort of the thing that we're sort of, or at least I'm sort of interested in, is how basic sensory processing can be modulated by the state that the animal is in. And whether that's previous experience with these tastes or fear or novelty or these kinds of things.
What are the clues in the connectivity? Because basolateral amygdala is kind of interposed in this. And so the hedonic value, which again, I mean, a lot of these questions are getting at like, the coding scheme, right? So there's, I guess, a lot of ideas about just temporal coding at a population level, and that somehow this signal is evolving more from, discrimination to making a choice based on this hedonic process, right? So how do we know what direction the signals are going? I mean, that's a stupid question, but like, do we have a sense of, is the hedonic input coming in, or is there sort of some cortical, is this an intrinsic cortical thing that then is sort of like?
So, well, I know from like conditioned taste aversion, right? So there's certainly maybe some of it is innate, right? There's definitely reciprocal connections to the amygdala, both bad and good parts of the amygdala, right? Into cortex. But I know there's more of a recent paper that looked, and they looked at responses in neurons, in gustatory cortex neurons before and after CTA. And they found that the majority of the cells that changed, this is anesthetized animals, but the majority of the cells that changed their response following the conditioned aversion were cells that I believe project to the basolateral amygdala, right? And so I think this is sort of getting to your question that, yeah, maybe there are these built-in circuits, right? And so some badness information is coming in from the amygdala into gustatory cortex when you learn the aversion. Although I think it's more complex than that too, because
Where, at what level in the periphery? I mean, there must be some, I mean, it's hard to believe that it's getting to cortex and then sort of having this exchange, because it's such a fundamental.
Right, and maybe that's the learning. I mean, the aversion, like sickness probably comes in the brainstem, right? And probably even PBN. I think there's evidence that PBN neurons also, encode badness as well. So it just gets really complex, and maybe, clearly the amygdala, if you lesion the amygdala, I think you block CTA. That's probably a safe bet, right? And so just like, so yeah, maybe, I don't know. It gets really difficult really fast. But yeah, it's clear that there's innate, and that, I think that's also hard because in the olfactory system, there is some, but there are much less innately bad odors. I mean, there are like predator odors and things, but the vast majority of the odors that we probably encounter aren't that bad, right? There are some, but not certainly like we have a whole taste system for like bad things, like a couple of bad things like bitters and sours, right? So it's just hard because there's a heavy innate, like you're saying, there's a heavy hedonic value. Sweet is good, right? And bitter is bad normally, right? And that's independent of any learning, right? So I think that makes it complex sometimes, because that's what I guess I was trying to get at today, is that if you're asking questions about how experience changes things, and you've got this extra layer of things are already good or bad coming in.
It seems like
in the olfactory system. Yeah, and so like
to follow the bad taste, to find the part of the brain that means bad. And follow the good taste, the part of the brain that means good. Shouldn't that be? I mean, it's really hardwired like that. Like bad is bad and good is good, and so bitter is always going to mean bad. Then let's just follow the bitter axons to the part of the brain that detects bad. Is there such a place?
I mean, maybe. Right, but I mean, it's also, but that's the problem, is some of the bad is happening in the brain, like like the other problem is when we think about like we have this reflex to if you pick up something and you don't know what it is and it's gross, you're spitting it out or you're making that face, and that's very, I think that's very, you don't need a cortex to do that, right? Babies do this, animals do, we all make that face, right? And so there's kind of different levels of bad, right? Maybe that's just the innate bad, but what about the learned bad, right? Maybe that is, I think, as most people probably say, the bad places in the amygdala, right? In terms of learned badness.
So can you do CTA without the gustatory cortex, or partly?
Yes.
So
Yes.
how much? Like, does it affect it a lot or just a little bit?
I'm trying to think. It's been done a number of times and with sort of mixed results. If I remember right, in the gustatory region, yes, right? But as you get further back into more posterior areas of cortex, maybe no. But this is again sort of fitting into this idea that maybe that's the region that encodes the sickness, right? So if you don't perceive the badness, you can't learn that it's bad. I think I want to say,
and that's one problem, right? Because all of this stuff, all this sensory information, taste, and malaise are all inputted into the same kind of region in the brainstem. So it's all funneling into this like NTS region that's picking up all sorts of stuff, right? So like that, I always find that amazing, like how much stuff goes through that small region of the brain and then goes to all these other,
right. And it's tiny,
circuits.
right? If you lesion that, you don't learn a CTA, right? You definitely don't. But then I think that sort of fits into this idea that, maybe thinking about gustatory cortex as its independent thing in insular cortex is just not really the way to do it. Maybe insular cortex is this area that represents all kinds of different body states and internal states. And one of those internal states is things in your mouth or things in your gut or these kinds of things. So, and maybe that makes more sense in the long run. And that leads you to, better experiments or more interesting experiments than sort of just, asking how similar two tastes are in cortex.
This always blows my mind, the gut stuff, impinging back on a learning mechanism, because the envelope of time that you have to integrate across becomes huge. I mean, I don't know how long it takes for a signal to get from the gut to the brain after eating something and having it make you, but for me, it might be hours. I don't know.
Yeah, and so that's what's crazy about CTA. So like, we do odor fear where we pair an odor with shock. And if you pair that, if you give 2 seconds of odor and then you wait a minute to give the shock, the animal is not going to learn that association, right? It's too far. But in CTA, it can be like the next day, right? I mean, it's like, that may be extreme, but it's certainly, you can wait, you can have an animal drink a bunch of saccharin or sugar and then wait several hours and then inject and make him sick and they still learn it, right? So it's different. And then that's the other thing is, in most of the studies, like what we use sodium chloride, but a lot of them use saccharin because saccharin is sweet, but doesn't have post-ingestive effects. So it's kind of hard, right? Like, because that also can confound things, right? So
it's always surprising to me that with this little bit of CTA that I looked at for the gustatory stuff, that the entire mechanism doesn't become aversive. Like they still lick, they still approach, they'll still try, like unless I'm not understanding something about the behavioral, the way things are blocked. But like you showed some stuff today that looked like the behavior has absolutely no effect, even after CT, across the board, it's similar to the control rather than the post-aversion training.
Yeah, and I think part of the issue is that, it's tricky because behavior gets tricky because you have to make the animal do what you want, but that's another manipulation. So like in the CTA experiments, to get the animals to drink, they have to be thirsty. So they're water deprived, right? And so when you put them in the next day, they have to drink. They haven't had water in a while, right? And so you're playing this game. I think I always think of if we were stuck out in the desert for a day or two and we didn't have water, if you came up on a big muddy puddle on the ground, you would just drink it, right? You're like, I don't care that this is going to taste terrible because I need water, right? And so there's all these other things that matter. Whereas that if I had a bottle of water in my hand and you had the choice between the muddy water and you would take the clean water, but you kind of have to do what you have to do. So all these things are tricky, right? Behavior is sometimes hard, and that's what I think I like about it. But it's also I think a lot of times you see behavior done and it's like, well, I did this behavior and here it is, right? And you didn't really think about like, what is the animal really telling me here? And sometimes even in CTA experiments, the first time they drink it, they may drink it very similar to water, right? And then as they have more trials and they sort of figure out, okay, because they don't know what they're getting that day. Maybe they, I'm sort of thinking for them here, but they don't know. If they go in and they're like, oh no, this is the stuff that I don't like, it made me sick, but maybe this is all I'm getting today. I guess some decision has to be made on some level in the system of whether or not just go ahead and drink it or avoid it, right? And then maybe as they get alternating or randomly alternating trials of water, they sort of start figuring out, okay, I'm gonna get water too, so I don't need to drink these other things.
Given the delay and the conditioned taste aversion, I thought maybe I ought to say what CTA stands for. Given the delay and conditioned taste aversion, it's amazing that it's ever very specific, because a lot of things happen in that delay. So I ate the potato salad. I also had a Coke, and then I got food poisoning that evening, and I blame the potato salad somehow. So I unconsciously, I mean, I develop an aversion to that.
And partially, I think it is also the novelty thing, right? So like you've had Coke a
Yes.
1,000 times and you've
That's
never
a-
gotten sick from Coke. So your Coke is a safe bet, right? Potato
Also,
salad.
you think there's a lot of logic, and so I had this idea that conditioned taste aversion was so primitive that it didn't involve a ton of logic and this sort of probabilistic thinking about my past experience. But is it like that?
Yeah.
But that's what Max shows in this kind of this idea of the latent inhibition thing, which I found was truly fascinating.
Latent inhibition is crazy. So if you give an animal sodium chloride for 10 days in a row, and then on the 11th day you switch it and give them lithium chloride and they get sick, they're not going to learn a taste aversion, right? Because it for whatever reason, and so people have couched this as that it's not novel. You need to learn it you only really learn a CTA to something new, right? And then look at this as a word related to acetylcholine, because acetylcholine comes in when it's novel. But it's a crazy thing. Yeah, this idea
and it perfectly relates to the Coke versus the potato salad.
Yeah, that was a really good analogy. Yeah, that's latent inhibition. Yeah, so maybe if you would have had some different drink that you had never had before with the potato salad, you may be like, oh, I don't really know which one made me sick. Maybe I should just avoid both. But that's a great latent inhibition analogy.
So maybe that's what gustatory cortex does. I mean, because you could potentially look at the relative disruption of latent inhibition versus just normal CTA with or without gustatory cortex, right? So that's to me that sounds a lot more like what cortex does, right? It decides what's new and whatever. It's not just the things that are there. It's really learning about their different.
Yeah, and so that has been done. I'm not sure about the lesion study, but certainly acetylcholine is heavily involved in obviously the novelty of the new stimulus. And if you block acetylcholine in gustatory cortex, you can disrupt learning, but you can also block latent inhibition by adding acetylcholine back in, and then therefore the circuit sees this once familiar stimulus as novel, because now there's this novelty signal tied to it, and then you will learn a CTA, right? So that people, the sort of it's weird that the taste system coming from the outside is a little bit weird, because you have this group, that's sort of small group of people. There's not very many olfactory people, but there's even less taste people, and they're really interested in truly taste coding and taste processing. But then you have this other group of people that use CTA as a mechanism for understanding learning, and all the things that go along with it, and they don't often communicate, right? And so there's like a ton of CTA stuff where you'll just see people who are doing taste CTA, but they don't care anything about like the taste coding aspect of it. It's all just about the circuits underlying that specific kind of learning. And so it's always been weird that there's this sort of disconnect between that. We don't talk to each other, and I'm sure we should, but we probably all say that and then don't.
So I think we lost Charlie, but we can't really turn his camera off for him. But you mentioned neophobia a few times, and that seems to be related to expectation, which I wasn't expecting to see in a lot of this stuff on a cellular level, right? Not that, anyway, but I'm just wondering if there's any parallel to that in olfactory cortex, because there are sort of anticipatory things happening in somatosensory and visual cortex, but I'm struggling to understand how neophobia works, because that seems very top down, and I don't really, can you just explain? You mentioned it a few times and I'm not sure.
Yeah, so I mean, I think
inability to approach, right?
Yeah, so neophobia can probably mean a lot of things depending on who you are, but so from true taste neophobia, the idea here is that the animal has just gotten water every day, right? He goes up to the little spout, I throw him in there, he knows when that shutter opens, it's water. And then on some day, after a whole bunch of water days, there's something new there, right? And I think the canonical explanation of that is it's sort of all couched under the same thing as CTA, where I don't know if this is going to be safe or not, right? And so I'm going to drink a little bit of it, but I'm going to be hesitant. I'm not just going to pound it, even though maybe it tastes good, because I don't know if it's poisonous or not, right? And so that's neophobia, right? My consumption is not what it would be. It's reduced, right? And then the next day I wait and I don't get sick, right? And so then I realize that okay, well, that must be safe. And so I'm going to drink it. And not only is it safe and I'm going to drink it, I like it, so I'm going to drink a lot of it, right? And then you've sort of attenuated this neophobia, and that's sort of what we see, right? And so some people, I think, sort of tie them together, where it's all kind of one sort of thing where you do, but it's probably true for other things. I mean, neophobia may not be the right word. I don't know. I mean, we probably all do this, right? You kind of change your behavior when you encounter something novel in your environment. Mice do this, right? If you throw something in their cage that they've never had before, they go through all these behaviors that are different than their normal right? They may sort of do some sniffing where they kind of go out and come back, right? There's sort of some risk assessment behaviors, maybe they're called, right? So I think it's sort of the same thing as that, where you don't really know if this is a safe or not safe thing, and it's crazy because the only way you can know if it's safe or not safe taste is to try it, right? So it's different than just looking at. If I see something on the floor that kind of looks weird, I can be like, well, I don't know, but I can see it from here, so I'm good. I don't have to go over there and lick it to know if it's safe or not.
Which,
so I think
kind of goes back to your earlier mention of, we kind of have to force these mice to behave the way, to actually do the experiment. We have to water deprive them, which is rough, right? They're in a state of thirst, so they're forced to go overcome their neophobia and at least sample the spouts. And I think this and as you mentioned that before, I kind of brought up an idea of do you ever plan to do these kind of like mini scope analysis on freely behaving like over multiple days and like in home cage environments or something that they won't have to be thirsty all the time in order to actually do
right.
experiment. So
you're right. That's a great point. So we have to sort of force them into this paradigm, because I want these little discrete events where I know when they're sampling and when they're not. But the other old way of doing it is I just take two bottles, like after I've made them sick, the next day you get a bottle with sodium chloride and you get a bottle with water, and it sits in there for a day. And then the animal, he's going to take a little bit, he can sort of do what he wants over time. And that's probably more naturalistic, but traditionally recording for that long is a lot, right? And I think we're sort of dependent on the people who build these things, the technology to do it. But I know there's the newer miniscopes, they're talking about doing these like 24-hour a day recordings. But I can't imagine the processing power. I mean, you have to know, it just gets out of hand quickly. I mean, you have to know everything the animal is doing, and then you have to it's a gigabyte a minute recording right now, right? So, I mean, even if you somehow dealt with that, you just, maybe it's possible, but it's just so overwhelming to even think about, like, oh my gosh, what if I had 24 hours worth of record? I mean, my computer can't even open up an Excel spreadsheet of the traces for 40 minutes. So, it just gets crazy big data, but that's cool because there's so much stuff there.
So if we go back to the original idea, like, is this a feeding area versus a perceptual area? Even just defining feeding behavior, feeding is so much more than taste, right? Like, 'cause you mentioned foraging in some of your reviews, right? I mean, what is there a role for this swath of cortex that's getting all this input from taste thalamus in foraging behavior that has nothing to do with taste sense? Like, is there some sort of larger distributed set of behaviors around feeding that we can think about in terms of the cortex?
I mean, I don't know. I mean, I guess if we think about it contextually, what the animal is doing, right? I mean, I guess they're just wandering around, smelling things, and if it smells safe, you may take a bite out of it and see. So it's hard, right? I mean, it's hard because for us, we use it in a very different way than, say, the animals are saying. Same for olfaction, right? They're not using it in the same way we are. So it's sometimes hard to, I think it's hard to really put that together. But, like they have to actively lick, right? And so this is sort of an interesting part of that. Like you can try not to smell things, or you can try to smell things, but generally you are smelling things, right? And you are seeing things and hearing things, but you're not tasting anything unless you taste, you have to physically do that thing. So it's a little bit different, right? I mean, you have to actively sample to get the information, which is, I think, is a little bit different than became very apparent as we've done similar experiments where you can just blow odor on the animal every day and see how I would call that passive experience, right? You don't really care about it, it's just happening. But then when I was making this up, I was like, oh, I'm going to call this passive experience. But then it occurred to me, well, it's not really passive at all, right? They're choosing when to lick, and that's a whole other sort of level of complexity, that goes into these things that makes it hard.
So where is the field? I mean, you mentioned the field is super small right now in terms of cortical gustatory cortex. So where do you see things going? What are the next round of big questions? Is it this naturalistic behaviors the way most places are headed?
Yeah, I think so. I mean, I think that like as we get better at being able to do things like record from longer times in the freely moving animal, you can really ask questions more related to what the animal, not just these forced things. I mean, I think back to like, originally we were doing, even in grad school, I was just taking an anesthetized animal, putting a single unit electrode in, recording from maybe 2 cells if I was lucky, and blowing some motors on and asking what happened, right? To now we've moved to, sampling from lots of cells in an awake animal too, but that animal was head fixed. So then the animal could only do a few things, to now we're moving to, freely moving to ultimately like sort of what Lindsey was talking about, to maybe we can just do the whole thing, we can, and then correlate those to behavior. So maybe that's the way to go about it, is if I can look at, and what I didn't show today is that we have a little bit of data that maybe, and there's some evidence up there that a cool idea is to look when the animal gets sick, right? Because the animal's drinking this sodium chloride and he thinks it's actually lithium chloride, but he thinks it's just salty. And then over time, you start getting sick, and he starts slowing his sickness down, and they kind of just they look really sad. They just like flatten out and lay on the corner, and it's clear that they don't feel well, and then they bounce back and then they're normal again, right? And so, if you think gustatory cortex is involved in this somehow, maybe you can see correlates of the learning happen sort of in real time in the animal. Like, does the activity in GC sort of correlate with, the sickness of the animal or the behavior in that way? And that's what I really like about the mini scopes, is that it's fairly easy to do that. In a way that I don't think you could do with other approaches. There's obviously drawbacks to using it, but I think all approaches have drawbacks. But I would hope, that's at least I would hope, is that we start maybe thinking a little bit bigger and
in terms of rhythms too, right?
Yeah.
Larger. It's always so much fun talking about taste, because it's so visceral and intuitive for us. But any last questions, guys? Because we're running a little low on time. We have 15 minutes left, but thank you for being with us. Otherwise, anyone else? No? I have to think of a better sign off. I'm not good at the sign off part because I just never want it to end. But thanks for joining us. This has been really great, and we're looking forward to seeing more from everyone in the taste field and learning. I mean, Lindsey's been bringing some really cool people around to talk to us about the explosive and just incredible things happening in this area, in the gut, as well as now the cortex. So thanks for doing this, guys, and we'll see you next week. This has been Neuroscientists Talk Shop.